All right, welcome back. Today we're going to talk about the M phase. So the M phase has, M phase is metaphase, right? And there's four distinct parts: prophase, metaphase, anaphase, and telophase. But prophase, there's enough happening. Most people now break it into two parts, right? So there's a pro-metaphase. So there's a pro-metaphase happening between prophase and metaphase. So let's get into it. All right, so in prophase, the chromatin, so DNA, right? So we all picture like that classic chromosome, right? It's connected in the middle here. Well, that's not a chromosome. This is what's called a mitosis chromosome, right? But before chromosomes are condensed and paired, they're kind of like spaghetti, right? And if you could look at them even further, we'll talk about later, they're wound around proteins called histones, right? And then your here, and a little lining there, a little lining here, and a little lining there, and it still goes on. So chromatin is the form of uncondensed chromosomes, right? So if you hear it say that, that's what it means. So chromatin begins to tightly coil or condense, right? So our spaghetti is kind of condensing to become that H-like structure. The nuclear envelope begins to disappear. Remember, it's disappearing because MPF is now starting to get up toward its peak. And the MPF, remember, is the thing that takes apart the nuclear envelope, right? Removal or disassembly. Right, so it's that MPF. The nucleoli disappears. Remember, that's where we get our RNA from. And each chromosome is now a complex of sister chromatids. So if we have our condensed right here, these are sister chromatids. And what those are is they're replicated chromosomes, right? So this one was made from that one, okay? And there's a couple things, right? So these are sister chromatids, and they're attached at the centromere. So they're attached at the centromere. And the centromere is that place in the middle where they're attached. Okay, so that's the centromere. The mitotic spindle begins to form. So if you look at these pictures over here, look at that top picture, right? So I have my centrioles that become centrosomes. Right? So these are centrioles, right? And they become so centrioles are organelles, and when they get those spindle fibers, they become centrosomes, right? So they start to grow spindle fibers, and then they start to move away from each other because they want to move toward the opposite sides of the cell. Right? So they want to eventually get here to the opposite sides of the cell. So they move away from each other, and then pro-metaphase, right? The nuclear envelope completely fragments. Like you have enough MPF, and it's attached, and it's taking the whole thing apart. And the microtubules now are moving through the center of the cell, if you can see that bottom picture. And they'll find what's called the kinetochore, right? So the kinetochore is in the centromere. So if you see in the bottom picture here, right, if you see those little green things, that's the kinetochore region of the chromosome. So they'll find that kinetochore and they'll attach. Okay, so that's the centromere. So if we look at our picture of our onion cell here, right? This picture right here, this is what we hope to do, but we're in distance learning, is that's an onion cell, right, that's in pro-metaphase. Okay, you can see the chromosomes condense. The nucleus is still a circle, so it still has a little bit of nuclear envelope in it, and it's getting ready to do what it's got to do. Okay, so let me try to condense this again. There we go. All right. All right, so now we're going to metaphase, right? So here's the thing. Metaphase is the longest phase of mitosis. Now don't confuse this with, remember that interphase is the longest phase of the cell cycle. Well, metaphase, right, is the longest of mitosis. Now, meta means middle. Alright, so meta means middle. I don't even like the way I wrote that. So meta means middle. Alright, so centrosomes are now at the opposite side. Remember they're centrosomes because they have the spindle fibers coming out. They're opposite sides now. The chromosomes are not the metaphase plate. So this in the middle is the metaphase plate. Remember, this is where I'm going to have my checkpoint, right? So metaphase plate is where my checkpoint is. So the checkpoint is to make sure that all the DNA is lined up, right? Chromosomes are now on the metaphase plate. Kinetic core of sister chromosomes now have 2 spindle fibers attached to them, right? A spindle fiber from each side, right? And then the microtubule complex is now called a spindle, right? So the centrosomes made these fibers. Now this is called the spindle, right? And like I said, if you look at this picture at the bottom, this is an animal cell, and this is metaphase because all the chromosomes are lined. And you can, if you look closely, you can see the spindle fibers there as well, right? There's no nucleus anymore. And we're getting ready to go. All right. So now we're at anaphase. So anaphase is the shortest phase of mitosis, right? So ana is away, right? So this is the phase where they go away. So here's what happens. The spindle fibers, sister chromosomes are pulled apart as spindle fibers shorten. So the centrosomes kind of reel back in those spindle fibers, and they shorten. And as they do, remember they're attached to the kinetic core region, they pull them away. Okay? And they migrate, or they separate toward the opposite sides of the pole. So each one of these is moving in a direction toward the centrosome. At the end of the anaphase, the polar regions have equivalent amount of chromosomes because they pulled each one in half. Right? They pulled each one in half, and all the DNA is the same on each side because they were all copies of each other. And you have telophase, right? So tele means away or far. So here we go. So remember, if I go back up here, so an important part of here is MPF is deactivated. Remember, MPF is holding those pieces of the nuclear envelope, right? And so they can't reattach. So it's deactivated. So as we get into the other phase, the nuclear envelope begins to form. 2 daughter cell nuclei begin to form because that nuclear envelope, the MPF, is almost all gone now. And so that nuclear envelope, when it finds another piece, it just locks together, right? And they continue to lock together and form a ball, right? So the chromosomes are now at the separate side, right? The nuclear envelope begins to form. Chromosomes become less dense, so they're becoming chromatin again. And then mitosis at the end of telophase is complete. So if you look at this, right, so that cell down here, that's at the end. Right? So I've got 2 nuclei, and you can see I've got this border of a cell beginning to form, right, that overlap. So there's a border of a cell beginning to form. Right, so mitosis is now complete. And then cytokinesis. So cytokinesis is, a lot of people put it in mitosis, but it's not. Right? So it's after mitosis. It's before mitosis and interphase. So if you think about it this way, cytokinesis is the border of mitosis and interphase. Now cytokinesis doesn't happen after mitosis. It starts during telophase. Right? During telophase, it's already preparing to split, and at the end of telophase, it splits. Right? And so what happens is 2 things. For animal cells, we get what's called cleavage furrow. And the cleavage furrow is the plasma membrane starts to pinch in on each side. Right? So that's the cleavage furrow. Right? And it will continue to pinch until it pinches the whole way, and then it'll separate into two cells. Right? Remember, cells have cell walls, and we don't. Right? So the cell plate, right, begins to form. So if we look at this picture right here, you can see the cell plate is beginning to form right there, and it will pinch it in half. Right? And then we'll have, like this is one that just finished. Right? So the cell wall will pinch it in half. All right, so that is what we had to get through today. Not very long. And